My name is Mallik Sundaram. I am the President and CEO of Mitra Biotech. We have offices in Bangalore, India and Boston, Massachusetts. Precision medicine has taken a big role over the last 10 years. Largely it has two different approaches. One is genomic approaches, where we look at patients' DNA or RNA. Based on that thing, make a prediction as to what drug is going to work or not going to work, and also risk stratification as to what patients fall into the higher risk category and what patients go into the lower risk category. Then the other approach is the phenotypic approach, where on a functional basis it's decided as to what drug is going to work or not going to work. And globally there are multiple companies. You have companies such as Foundation Medicine and Genomic Health in the US, among the more well-known companies. And in India you have companies such as Stand Genomics, MedGenome and others. Well, Genomics has actually ridden on the lot of data that pharma has developed on biomarker companion diagnostic application, and over the last 10 years or so, people really thought that this is going to be the ultimate solution that everybody is actually looking for. But what has happened over the last 10 years, last 5 years, is that people are becoming increasingly aware of the challenges with the system. So what happens is that you take a DNA, RNA, and first is like you find out what are the actionable mutations, and what happens is that very often it's only about kind of 50% of the cases where you have something called an actionable mutation. And then for those actionable mutations also, it's not as if the drugs are readily available, right? So there are drugs that are commercially available, then there are drugs that are just in the developing phase. So what it means is, for the patient under consideration, he can only be given what are the drugs that is available. So you have you eliminate a large number of patients, 50% of the patients, based on the absence of an actionable mutations, and then you eliminate another 40% of those patients based on the drugs that are commercially available. And then further you also you also talk about among these actionable mutations and the available drugs, what drugs actually kind of work on the patients. So if you do all of this various kind of the selections, ultimately what happens is that it's actually 1 or 2% of patients on whom genomics actually plays a very beneficial role for the patients. Add to this the large classes of drugs for which genomics does not have an answer. You have this newer generation of immune checkpoint inhibitors where genomics cannot be used, and then also the older generation of chemotherapy, which is still a mainstay in cancer treatment. So genomics cannot play a role on that also, right? So there is a kind of a class of drugs, targeted drugs, for which genomics definitely plays a role, but on that how many patients actually benefit, it's become a questionable one. So the alternative option is phenotypic approaches, and that also has its own challenges, right? So what phenotypic means is that you take a patient's tumor cells, manipulate it in some way, and ask the question as to whether the given drug is going to work or not. So historically what has happened is people have tried to kind of make a very simple solution for a heterogeneous disease such as cancer. It is something like, I lost my key, but I am actually going to look at it under the street light because that's where the light is, right? You are obviously not going to kind of find it. So like that, because people are looking at overly simplistic models, that approach has not been kind of successful so far. But over the last few years, what companies such as Mitra Biotech is doing is that we are adapting heterogeneous approach, complex models to address this complex problem, and we have a tremendous amount of data to show that not only is phenotypic approach, if done in the right way, is going to be beneficial to the precision diagnostics, but it's actually going to be kind of complementary to some of the findings that genomics actually is good in solving. What we do in Mitra Biotech is we have developed a technology called CanScript. What at fundamental level what it does is, what it is based on is, it's based on the concept of recreating the tumor microenvironment on an in vitro system. It is done on a multiple levels. It is almost like an onion, right? So at one level it is personalized to the type of cancer, to the subtypes of cancer. So we have specialized plates where it is coated with the tumor matrix proteins that is specific for let's say kind of a breast cancer and a colon cancer. Not only that, we have a specific plate that is applicable for triple negative breast cancer, hormone dependent breast cancer, and so on and so forth. The second layer of personalization comes from the patient's own blood. What we do is that in addition to collecting patient's tumor biopsy, we also collect autologous blood, and from the patient's blood we isolate serum derived ligands are used. We also isolate immune environment. So what each of this does is that it adds additional level of personalization. So the serum ligands, for example, is that when you put a drug in the patient's tumor, it actually provides the appropriate ligands so that the drug is tricked into thinking as if it is within the body. So by providing these various personalization, and then also the other improvisation that we have done is we don't look at just a single metric, single output such as the cell death or cell proliferation. So the way that we look at it is that it's almost like an elephant. You cannot describe an elephant by an eye or a ear or a tail. What you need to do is that you need to look at each one of these measures, and then you need to provide a composite picture of that. In a similar manner, what we do is that we look at cell viability, cell proliferation, cell death. Cell proliferation, we do multiple metrics, such we do the standard Ka67 type of an assay, but we also do other things such as, okay, if the tumor cell is growing, what type of a pathway that it takes. Does it take ATP, does it take ADP, glucose, and so on, right? So there's a lot of science actually goes into this. And what we also have is that if I take each one of these kind of a 15 or 20 different metric and present it to the doctor, it's not going to really kind of give a solution, right? So what a physician is looking for is that for my patient, how can you provide a simple solution, right? So what we have done is that we have taken a complex approach, but we have also taken technology. We have used bioinformatics algorithm. We have also taken a tremendous amount of clinical data set on the same patients on whom we have done this CanScript analysis. By using a combination of all of this, we are able to come up with a predictive score called M-score. Higher the M-score, higher is the chance of a patient responding. And one thing to be kind of noted here is that our technology is applicable for chemotherapeutics, it is applicable for targeted drugs, it's also applicable for the immune checkpoint inhibitors, the latest drugs that are being kind of developed. And not only this, we have developed it in such a way that there is a minimal amount of time that we spend. So from the time the tumor is excised from the patient's body to the time a report is actually sitting in the hands of a patient of the physician, the total time is about 7 days. CanScript as a diagnostic application, it has been launched in India, and later part of this year we would be launching it in the United States. Besides diagnostics, we have a very strong presence in the biopharma applications, where we help biopharma companies as they strategize, as they come up with their clinical development strategy. CanScript becomes an invaluable tool in deciding what kind of cancer is their drug applicable, what kind of combination strategies that they do, and further to the level of what kind of a companion diagnostics can be developed. So in all of those various applications, we work with various global biopharma companies, and I am also glad to say that to date CanScript is the only technology that is beneficial on the immune checkpoint inhibitor development. So we are developing additional clinical data set on that field also, even as we are speaking.
Right. And tell us something about the ongoing studies.
This is one field where you cannot just stop that we have done everything and we are launching it commercially. The two things need to kind of go hand in hand. And Mitra as a company strongly believes in clinical validation, clinical data, as well as in demonstrating benefits to the patient. To that effort, we do various studies both in India as well as outside India. So some of these studies are done as an investigator initiated studies, other studies are done as company sponsored studies, and later part of this year, in the next few months, we will be announcing the studies that we are doing in India as well as in Europe and the United States.